Evening, everyone, and welcome to our live stream for Paper 2 Biology. So the goal of tonight is to go through everything that could come up on the exam paper tomorrow. This is, as always, OCR Gateway specific, but I'm sure there is overlap with certain aspects of the other boards if that's what you're doing. Obviously, if you're OCR Gateway, perfect, this is for you. We are going to go through all of the separate sciences, so the separate biology and also combined science, foundation and higher tier content, so the whole lot. Usually this one takes around about an hour and 45 minutes, so we shall see how we do tonight. We do have the chat going, so use that obviously to post any questions, anything you're not 100% certain on. I will try and get to it, but if it gets filled with general rambles, then there is always the chance that I can miss it. So let's all be polite and sensible, and let's go through some biology then. Okay, so first one, tomorrow's paper is going to be units 4, 5, and 6. So B4 is basically all about ecosystems. So first thing we need to know then is a few key terms that could crop up here. When we're talking about an ecosystem, what we're actually referring to are all of those living organisms and the physical conditions in a given area. If we talk about the community, those are all of the organisms within that ecosystem. The habitat, where the organisms live, should have known that from Key Stage 3, I hope. Producers, this is the organism that is able to make its own food, usually through photosynthesis. So we're looking at things like your plants and your algae at this point. We then have the consumers, which are the ones that can't make their own food. So in order to actually meet their energy demands, they have to eat some kind of other organism. So these are your typical animals at this point. And then we've got our decomposers, which are the ones that are going to gain all of their energy requirements through feeding on either dead or decaying matter. So for those of you doing just the triple science, the biology only, then you need to know a little bit more about the detail to do with our food chains. So hopefully we remember the basics of our food chain, that when we're actually writing them, obviously it's showing the flow of energy or biomass through a given system. Now, they could ask you here to draw on some of that knowledge that is now assumed from your paper 1 content. As we've said, producers at the start of the food chain are making their own food through a process like photosynthesis. Now, photosynthesis was a paper 1 topic. That doesn't mean that you cannot be asked things like the word or the balance symbol equation for photosynthesis here. So just a reminder, carbon dioxide and water makes glucose and oxygen, or in terms of our symbols, 6CO2 plus 6H2O makes C6H12O6 plus 6O2. So make sure you can do those because they could always come up on the exam tomorrow. When we've actually created that glucose, it doesn't just stay as glucose forever. We actually convert that glucose into a range of other materials. Some of it, for example, get converted into the cellulose for the plant cell walls. Some of it into carbohydrates and proteins. All of that is going to make up this biomass, the living material within the plant. Then when an animal eats the plant, that gets transferred from one organism to the next, but not all of it. Remember, some of that biomass is lost at each stage of the food chain. So when we're actually drawing out our food chains, hopefully this is a really obvious one, that when we're actually writing it, obviously we put down the organisms and then we are going to put an arrow between them. Do remember the direction of the arrow is important. You can't just draw a line, you need to have an arrowhead, and the arrowhead needs to be pointing in the direction of the energy transfer or the biomass transfer. So what we're seeing here is that we start with our substances in the grass and it moves to the rabbit. If you had the arrows going the other way, what you'd actually be saying is that that biomass from the rabbit is going to end up in the grass, which basically is you turning around and saying that rabbits that are happy hopping around in a field all of a sudden are going to have a whole pack of grass leap out from behind a hill and devour them, leaving nothing but a skeleton behind. Doesn't happen. At least not yet. So arrows are important. There is also a phrase that we could see here called a trophic level. Now, a trophic level is just a feeding level. So we would mean a trophic level, the producers, the primary consumers, secondary consumers. So what we're really doing is looking to see what they're actually doing. Now, in a food chain, it's dead easy. Start with the producer, next one along is the primary consumer, and so on. You could also be given a food web where they're all interlinking food chains. You just have to do a little look here. Start at the bottom where we've got the organisms that are known, the plants. They are always the producers. Then follow that arrow to the first organism you come to. They will always be the primary consumers because primary consumers feed on the producers. If you follow the arrow from the primary consumer, you're going to hit a secondary consumer. So you can just work your way through any given food web nice and simply. Start with the producer and then work your way up. Primary, secondary, tertiary, all consumers, different levels there. Right, back for everyone combined and triple, we need to know about biotic and abiotic factors. If we're talking about a biotic factor, these are the living factors. So think about competition between organisms here, okay? Something that's living that is going to therefore cause interactions in this environment. The abiotic factors are the non-living ones. So this would be your temperature, your pH, the light intensity, water availability. All of those are not living, they are abiotic factors. You do need to be able to give a little bit of an explanation about how those factors affect the organisms that live there. So light obviously is going to be required for photosynthesis. So more light, more photosynthesis, the more successful that plant will actually be. However, that's not to say plants can't grow in these lower light intensities. Some of them have adapted to grow in shaded areas. They've generally got really large leaves. Think of things like rhubarb, huge leaves because they're designed to grow in shady zones. Temperature is our next factor that we're going to consider. And this just ties back into the idea that we've got a whole range of enzyme-controlled reactions within our living organisms. Temperature gets too high, enzymes denature, reactions will therefore stop. Okay, if they warm up just enough, then obviously it's going to speed up those reactions because the enzymes are going to have more kinetic energy, more likely to collide with the substrate, and therefore more likely to form those enzyme-substrate complexes. So think of the examples there of things like your lizards, the little ectotherms, the ones that don't actually have a mechanism for warming themselves up internally. They have to rely on basically sitting in a sunny area. So they go and lie on a hot rock first thing in the morning, warms up their body, gets their reactions going because their enzymes gain that kinetic energy. Water availability is all down to the simple fact that if you don't have enough water as a living thing, you're going to die, okay? If we look at our plants with a lack of water, then the first little sign that there's a little bit going wrong there is that they're going to start to wilt. So their leaves start to droop, the whole thing goes a little bit sad. At that point, those cells are no longer turgid. They're not going to be able to carry out things like transpiration fully. When we come on to pH, then what we actually find is that's all to do with affecting what minerals are actually available in the soil to those plants. And different plants grow in different pH soils. So what we actually find there is that some plants are going to grow in acidic soils and others will grow in alkaline soils. And if you try and grow the wrong plant in the wrong soil, generally it does pretty badly. Quick reminder about the different ways that we can measure those abiotic factors, because this could tie into one of those sampling ideas. If you want to measure the light intensity, we use a light meter. If you want to measure the moisture that's present, it's a humidity sensor. pH is a pH probe, and temperature, hopefully the most obvious of them all, is a thermometer. So you can obviously talk about the bits of equipment we would use to measure any of those abiotic factors there. The next little section that we need to know about is the competition and interdependence bit. If we're considering having a number of organisms living in the same space, then they're going to have to compete for certain resources. For the simple fact, there's not going to be a never-ending supply of everything. So what we actually find is that in those instances where there is competition, those that are best adapted are going to get the resource. Those not as well adapted are not going to get that resource. So they have basically two choices. One, go somewhere else, or two, die. So most organisms are going to go somewhere else. If they can't, obviously that leads to a problem, and that's where we start to see the risks of extinction, etc. Now, when we talk about the population within this area, that is just the number of organisms that are living within that area at that given time. And what we find is this competition that's going on for resources will have a direct impact on that population size. So if we've got limited competition, then we're going to see populations increasing. If there's lots of competition, then some kind of population is going to decrease. Common sense, hopefully, on what plants and animals are competing for. As far as animals go, water, food, etc. Plants, we're looking at water, light, all of the usuals. We did mention this word interdependence, and this quite simply is how all these different organisms that live within a community are relying on one another, hence interdependence. They're all depending on each other. And we've got 3 ecological relationships we need to know about as far as GCSE goes. First one, the easiest one of all, I do believe, is our predator-prey relationship, so the predation. Now, this graph, I'm hoping, is rather familiar to us. So what one we've got here is 2 different colour lines. So the red line that goes higher, that is our prey, and then the blue line that's lower is the predator. They could ask you to either describe or explain this graph. If it's a describe, it's a say what you see. So what we do there is we'd say that initially we get an increase in the population of our prey animals, and then a short while later, there's an increase in the numbers of predators. When the prey animals decrease, a little while later, there's a decrease in the predators. That's a description. We've literally said what we see. If it was an explain, we do that first of all, say what we see, but we then need to say why that happens using some science knowledge. So what we do is add in where we've got this increase in our prey animals leading to that increase in the predators because there is more food available to support a larger population. Then, as that population of our predators increases, they're eating more of those prey animals, leads to a decrease in their population. So we're then going to see a decrease in the predators because they're running out of food. One point to note is that we do need to mention the fact that there is always a slight delay between the change in one population and the change in the other. It takes a while to either breed or starve to death at the end of the day. Second relationship we need to know is one called mutualism. If we are talking about a mutualistic relationship, this is where both of the organisms are going to benefit. So both of them are getting something from that relationship. Think of those little birds and crocodiles. So the little birds are obviously pecking all of the bits of debris from the crocodile's teeth, therefore making sure that their mouths are nice and clean. Meanwhile, the birds are going to be protected from these other random creatures that might want to eat them, because you know what? They're sitting on a crocodile. They're not going to go up to them and try and eat them at that point. Third relationship is parasitism, and this is where we have a parasitic relationship. So we have a parasite, we have a host. The parasite will benefit, the host will suffer. So the best example here is good old tapeworms. So what we see there is that if you happen to have a tapeworm, then it's going to be basically having a grand old time stealing all of those nutrients from your diet, and you are not going to be having such a good time because you're not getting any of those nutrients you are trying to consume. So not a great thing, certainly not a weight loss plan. So anyone who's trying to sell you that on the internet, don't do it. It's stupidity. Right, back for just the triple scientists now, the GCSE biology folks. We need to know about a couple of different types of pyramid. So first one is the pyramid of numbers. Now, pyramids of numbers, quite simply, the length of the bar tells us how many organisms there are, hence pyramid of numbers. We always start with the producers at the bottom and then just stack up those bars on top. If you're asked to draw a pyramid of numbers, pick a scale to draw it, if it obviously gives you a bit of graph paper. If it just gives you a big blank space and asks you to sketch a pyramid of numbers, then you don't need to measure, okay? You do, however, need each of those bars roughly showing an approximate length. So the biggest one, obviously, for the largest number of organisms and so on. Producers at the bottom, working further up as we go. Producer, primary consumer, secondary, tertiary, etc. Then label those bars. You don't have to squeeze it into the bar itself. You can put a little line going off to the side to give you a bit more room. One thing to note about our pyramid of numbers, they're not always pyramid shaped. bit of a lie there in the name. If you have a single tree, that is going to be a teeny little line at the bottom, and then we could have hundreds, if not thousands, of insects living on it. So the producer, teeny little bar, and then our primary consumers, quite large. So pyramids numbers, not always pyramid shaped. The second pyramid, which is the pyramid of biomass, is always pyramid shaped. So same thing, start with the producers at the bottom and then just stack those bars going up. And what we need to do is label those bars again, but this time it's taking into account not only the size of the population, but also the amount of living material that they are made from. So pyramids of biomass, always pyramid shaped. If they ask you how you would calculate the biomass to be able to draw one of these pyramids, then we would take samples from each of those trophic levels. We would work out the average mass of an organism from that trophic level, calculate the population size, and then multiply your average mass by the population size. That gives us total biomass for that particular trophic level. One downside to how we do this, we normally use something called dry mass. And this is where we basically removed all the water from the organism. Now, hopefully common sense is telling you that if we've completely dried out an organism, it's certainly not living anymore. So when we calculate dry mass, then we are killing an organism. So that's a bit of a downside of that technique. Still just GCSE biology, folks, we need to know that in terms of this transfer of the biomass and the energy from one part of our food chain to the next, it's not great in the grand scheme of things. We're actually seeing vast amounts of the energy that is starting off from the sun, going to those first organisms, our producers, not making it. It doesn't make it into that biomass. A load of it just reflects off the leaves. Some of it's used in the plant for things like respiration, etc. And therefore, at each stage of that food chain, biomass is going to be lost. So it's not overly efficient as a scheme. They could ask you to suggest some ways in which biomass is lost from a food chain. And what we've got are 4 key ideas here. First one, respiration. Every time we carry out respiration, we lose biomass. Second one is that we're not going to eat all parts of the organism. So it's not like when a rabbit eats a bit of grass, it digs up all of the roots to consume those. Not all animals eat every bit of a skeleton, for example. So there's always bits that are not going to be eaten. Third one is that we have the process of egestion, which is where we can't digest everything. So undigested material is going to pass out of the body as feces. And then we have excretion, which is where we lose waste products from the body. And that's the urine. So this is the pee and the poo part where we're going to be losing biomass from our organisms. Each stage, we lose a little bit. That does mean that our food chains are going to be limited in their length. They can't be any number of organisms. They're usually limited to 4 trophic levels, because beyond that, there's just not sufficient energy to support life. We find our calculation at this point as well, which is just an efficiency calculation. It's the efficiency of our biomass transfer. The way you'd work it out is mass after the transfer divided by the mass before the transfer times 100. So just double check that obviously they're in the same units, grams or kilograms makes no difference, as long as they're both the same. After divided by before times 100 to give you the percentage. Okay, back for everyone then. We're on to our fun-filled little cycles section. And there are 3 cycles that we need to know about. We need to know about the carbon cycle, and carbon is important to be cycling around our ecosystems because carbon is used to make fats, proteins, carbohydrates, DNA, name a biomolecule, you're good. Nitrogen cycle, which is used to make the DNA and the proteins, and the water cycle. Now, you'll notice that the carbon and the nitrogen cycles, we talked about what we need carbon and nitrogen for. Little way around it to minimize your learning is to avoid making mistakes and mixing them up. They both are used in DNA and proteins. So if they ask you to suggest what nitrogen or carbon is used to actually synthesize or make in a living thing, just go DNA and proteins because it covers both in one simple bit of learning. Right, let's start with probably the least favourite cycle, which is our nitrogen cycle then. Now, the nitrogen cycle always fills people with dread for some reason, but the reality of it is we need to know 4 microbes. That's pretty much the nitrogen cycle, 4 microbes and what they do. So, we have got decomposers, these are bacteria and fungi, and they are going to change the proteins into ammonia. So, decomposers take proteins and convert it to ammonia. Second microbe is going to be our nitrifying bacteria, and these ones change that ammonia into nitrates. Third one is the nitrogen fixing bacteria. These are going to take nitrogen gas from the air and change that into nitrates as well. And then finally, the denitrifying bacteria change the nitrates back into nitrogen gas. So this is what we've got as the cycle then. It looks worse than it is. So here's our nitrogen gas in the air. So obviously nitrogen is an inert gas. It's not something our plants can take up and utilize directly. So nitrogen fixing bacteria change the nitrogen into nitrates that can then be absorbed by the roots of the plant. We've obviously also got that ammonia being converted into nitrates by the nitrifying bacteria. And again, once we've got the nitrates absorbed by the roots of the plant and then it's incorporated into plant proteins. So once we've taken up the nitrates, plants make proteins. Those proteins in the plant can obviously be passed on to animals when they eat the plants. Our plants and our animals are going to die at some point, or they're going to excrete in the case of our animals. And that means that we're going to have proteins, etc., that can be broken down by the decomposers into our ammonia. And that is the nitrogen cycle. It's not terrible. 4 different organisms, decomposers, nitrifying, nitrogen fixing, and denitrifying bacteria. If you know their names, you know what they're converting, and then you remember animals eat plants, and that passes obviously the proteins from the plants into the animals, you've got the nitrogen cycle, and you can answer any of those questions on it. Second cycle then is the water cycle. So our water cycle has a few little P words on there that are maybe a little bit tricky to get our heads around. So let's start off, where should we start? Let's start over here with the clouds. So when we have clouds, eventually rain will fall, and that is a process called precipitation. So as the rain falls, it's obviously going to hit the land. Some of it is going to just run straight off and end up in a body of water. And some of it is going to trickle through little gaps in our rocks, and that is the process of percolation. When we've actually got the water in the ground, some of our plants are then obviously going to absorb it through their roots, and then, link back to our paper 1 stuff, our plants are going to release that water vapor to the air through the process of transpiration. So water vapor is going to obviously leave through those aerial parts of the plant, the leaves, and return to the air. We'll also have evaporation happening on any sort of standing bodies of water, and therefore obviously our water vapor is going to rise up. As it rises, it cools, it condenses to form the clouds, keeps cooling, and then falls as rain again in our precipitation, sorry. So what we've got there is our nice little water cycle. Make sure you know the names of those processes, because then you can obviously answer any of those key points on it. Third and final cycle is the carbon cycle. Now, this one is a nice simple one. Carbon dioxide in the atmosphere, first of all, there will only ever be a single arrow pointing away from the carbon dioxide in the atmosphere. That is always going to be pointing towards some kind of a plant, because it's photosynthesis again. So photosynthesis is the only process that removes carbon dioxide from the atmosphere. That carbon dioxide is then going to be built into the biomass of our plant, used to make carbohydrates, etc. And then we're going to see those carbohydrates being passed into the animals when the animal eats the plant again. We also have this process of respiration. You can see there's 3 arrows representing respiration there, which is going from the animals, from the plants, basically any living thing, back to the atmosphere, because respiration releases carbon dioxide. Paper 1 stuff again. Some things are obviously going to die, get buried underground, over millions of years turn into a fossil fuel, which we're then going to decide to burn. Good old combustion returns carbon dioxide to the atmosphere. So realistically, what we've got, photosynthesis removes carbon dioxide from the atmosphere, respiration and combustion are going to be returning it to the atmosphere. Now, it is worth remembering that our carbon dioxide levels do vary, and they're going to vary throughout the 24-hour period of each day because photosynthesis requires light. Therefore, if we've got nighttime, less photosynthesis. But it also varies across seasons. So obviously during the winter, we're going to have less photosynthesis happening and therefore a slight increase in carbon dioxide levels. You can find questions that do overlap with paper one content because at this point it's assumed knowledge. You've done paper one, where there's overlap, then they can drag across some of that paper one content. So hopefully you didn't hit reset on your brains as soon as you did that paper a couple of weeks back. One thing to bear in mind, though, is one of the major causes of carbon dioxide increasing in the atmosphere is, of course, humans. We are responsible for huge increases in the amount of carbon dioxide. We've seen that graph where it just shoots right up, all because of all of our activities, cutting down forests, burning loads of fossil fuels, etc. Last bit of B4 is about our decomposers then. When we're talking about a decomposer, these are bacteria and fungi, as we've already said, and they're going to be breaking down this dead or organic material into the constituent minerals, if you like, the bits it's made of, to return those to the soil. There is a phrase that sometimes comes up here called saprophyte. Now, this is just a description of how these organisms could feed. And saprophytes quite simply just release enzymes from themselves onto the surface of that dead material. It will then break it down. So we're going to actually digest that material. And then it just absorbs those individual little components, those nutrients. So if you see what does saprophyte mean, releases enzymes onto the surface of the dead material and then just absorbs the broken down nutrients. Second word we could see here is the word detritivore. Now, detritivores quite simply are small animals that assist in the process of decomposition. And they do this by actually eating and feeding on this organic material, and they're going to break it up into smaller pieces. So rather than having just one giant leaf, it's going to basically feed on it, break it up into smaller bits, and that increases the surface area for the decomposers to work on. Good examples here, things like an earthworm, for example, quite happily munching on all the dead leaves, breaking them up into smaller pieces, therefore increasing surface area. If you're doing GCSE biology, you do need to know the factors that can affect the rate of decomposition. This usually crops up as a really exciting question about a compost heap. So what we're going to find here is that if we've got warm temperatures, it's going to be a rapid rate. Too hot, enzymes denature. Too cold, reactions are too slow. We need it to be moist because the microorganisms need this damp environment in order to actually function. And we need plenty of oxygen. So look out for their typical questions where they've got compost bins with holes and without holes, or they're talking about turning over a compost heap. So basically digging a sort of spade into it and turning it. Whole idea behind both of those increases the oxygen, increasing the rate of decomposition, because all we're doing there, providing the oxygen, etc., that we need for respiration. Calculation could come in here, asking you to calculate the rate of decay. That's as simple as the change in mass divided by however long that took. So using a table to read it off, change in mass divided by the time in days. And that is unit 4. So next up, B5, this is basically genetics and natural selection. So first thing, variation. This is the difference that we see within a species. So usually we're talking about phenotypes, which are the observable characteristics. So think about eye color, hair color. Those are all phenotypes because we can see the differences there. If we're looking to see what causes these variations within our organism, then we've got two main reasons. First one, genetic, so the genes that are inherited from the parents. Second one, environmental, caused by the world in which you live. So obviously genetic variation, that's going to be linked into family. So whoever parents are, they're going to pass on their genes, which then means you're going to have those features. Things like blood group are a great example of this one, caused only by genes. We then have the environmental variation caused by the world in which you live. This is stuff that could change if you were in a different area or you had a different life experience. So accents, scars, tattoos, piercings, all of those are going to affect you as an individual, but they're not going to obviously be passed on to your offspring. I'm sure some people are very glad that things like tattoos and piercings are not passed on to offspring because you may not approve of what your parents have, for example. Then we do have some of these characteristics which are going to be both environmental and genetic. So these are where both of those are interacting to give us this extra variation. Good example there is height, because you may have super tall people in your family, but if you're not fed a decent diet with plenty of protein as you're growing up, you're never going to get to that maximum height. You're going to be a little bit shorter, I'm afraid. When we consider our variation, then we've got two types. We have continuous and discontinuous variation. So the continuous variation can take any value within a range, and these are going to be caused by both genetic and environmental causes. Normally, these are going to be controlled by multiple genes, not just a single one. There's going to be several others interacting here. So things like height are a great example of that. If they ask you what kind of graph we would use to show continuous variation, you need to say histogram. And hopefully remember, histograms are the ones without gaps between the bars. So all the bars must touch on a histogram if they ask you to draw one. We then have discontinuous, and this is going to give us very clear, distinct values. So blood type's a good example here. Your blood type's either A, B, AB, or O. So you're one of 4 blood types, that's it. These are going to be caused by either one or a very tiny number of genes, and it's only genetic. So if they ask you to plot a graph of a discontinuous value, it would be a bar chart. Each bar has a gap between the next one. This leads us nicely onto our 2 types of reproduction that we have available. We have asexual or sexual reproduction. If we consider asexual reproduction first, this is where we are going to be making clones, offspring that are genetically identical to their parents. Now, what we actually find then is organisms like bacteria reproduce asexually. So no sex involved, they're asexual reproduction, which is basically mitosis. We start off with a single cell, we replicate the DNA, then divides into 2 genetically identical cells. Remember, they're making diploid cells, 2 sets of that genetic material there. Some plants will also be able to use asexual reproduction. Most likely example you would have seen are good old strawberry plants or spider plants. Both do the same things. They send this little runner out, and then sitting at the end is a teeny tiny little cloned plant. We do have some weird little animals that also use asexual reproduction. So this is our starfish and our sea anemones. So for sea anemones, think Finding Nemo, where he lives. Sexual reproduction is where we are going to see 2 parents required. We need male and female to mix that genetic information. Because obviously we've got 2 parents mixing their genetic information at this point, then we are going to get greater variation within the offspring. And this is how most animals are actually going to reproduce, is using sexual reproduction. When we're looking at sexual reproduction, we have got 2 of these things called gametes. Now, a gamete is just a sex cell. So if we're looking at animals, sperm and egg, obviously we've got those 2 individual gametes that are going to come together and fuse during the process of fertilization. Now, we need to be able to make these gametes so that they can fuse to make an offspring. And the key thing about what has to happen for that is we need half the number of chromosomes in a gamete that we would find in a normal body cell. Watch out for these little questions that they throw in here about telling you some random creature and how many chromosomes it has in either a body cell or one of its gametes, and you're asked to work out how many chromosomes are in the other. So if they were to tell you a polar bear has 20 chromosomes in a body cell, how many does it have in a sperm cell? All you'd have to do is divide it by 2. There'd be 10. If they told you the gamete and they asked you about the normal body cell, just double it. Watch out if they're being tricky, because I do remember one year that they put a question on there talking about a skin cell or something, and then they asked you how many of chromosomes would be in a cell in the eye. They're still just body cells, so it's going to be the same number. So watch out for sneaky bits that they sometimes throw in. Now, plants also carry out sexual reproduction. So we've got the male gamete, which is the pollen, and an egg cell is the female. So obviously pollination is where we're transferring the pollen from one plant to another for that process of fertilization. Those of you doing the GCSE biology will need to know a comparison between sexual and asexual reproduction. So if we're thinking about asexual reproduction, then we only need one parent. Therefore, reproduction is faster. Benefit. We also will find that if the parent is really well adapted to the environment, all of the offspring will also be really well adapted because they are genetically identical to one another. The downside, though, is because they're all genetically identical to one another, if there is a change in one of those factors, either biotic or abiotic, we could wipe out the lot of them because they're all going to be equally susceptible to that change. For our sexual reproduction, the key advantage is we get variation. Because obviously we do have this mixing of the genetic material, there's greater variation, which means they can adapt to a greater range of situations. The downside, we need 2 parents, they've got to be able to mate, and therefore reproduction is much slower. Back for everyone again, we're going to recap on the process of meiosis, not to be confused with mitosis. Now, meiosis is the process where we are going to end up with 4 haploid cells. Haploid means we've got half the number of chromosomes as a normal body cell. So in humans, that would mean we have 23 chromosomes. What we actually find here is for the process of meiosis to occur, then it's going to start off identical to mitosis. First thing, our cell is going to replicate the DNA. So DNA is replicated first. Then our chromosomes line up along the equator, pull to the opposite poles, and then the cell divides by cytokinesis. Identical to mitosis as we've seen before. However, that's not the end of meiosis. We then have the chromosomes lining up on the equator again, pull to the opposite poles, and the cell divides. So at this point, we've now got 4 cells, and each of them has half the number of chromosomes as that original. So original cell, DNA replicates, chromosomes along the equator, pull to the opposite poles, cell divides. Chromosomes line up along the equator, pull to the opposite poles, cells divide. That's meiosis. It's basically mitosis plus another lining up, moving to poles, cells dividing. Don't throw in a second replication though, otherwise you just end up with more identical cells. Key thing about meiosis is it leads to variation, and we've got 3 key ways that we can get this variation. First one is what we call random assortment of our chromosomes, and by that what we mean is we've got obviously 2 chromosomes for each of them, and we don't know which way they're going to orientate themselves. So in each of those individual cells they could line up in a different way, hence random assortment at the equator. When they're all lined up with their little pairs very close, they can also have this process called crossing over, where literally they just cross over the chromosome in a little section and then they swap a little bit with each other. So they're literally swapping a little bit of chromosome with their neighbouring one. Third and final one is that random fertilization of the sperm and the egg. So it's completely down to chance as to which sperm will join with which egg. Okay. Now we come on to the little Punnett squares, working out how we inherit certain characteristics. Now, before we delve too deeply into this, we need to know a key term, which is the term allele. So when we're talking about a gene, that's a section of DNA that codes for a particular protein. Alleles are different forms of a gene. So you've got a gene for eye color. The alleles would be blue eyes, green eyes, gray eyes, etc. Okay, so each of those are different versions of that gene. Now, what we're going to find then is we need to be able to draw these typical genetic cross diagrams for a range of questions. If they're being nice, they will give you the outline for it with all of the individual alleles already set out. If they're not being so nice, then they could just give you a written context and expect you to work out what those alleles will be for the parents and then for the offspring. So to do that, we need to know some other words. Dominant alleles. Okay, when we're talking about a dominant allele, these are going to be capital letters on our diagrams. And basically, to show a trait that is dominant, you only need a single dominant allele present. So if you've got one capital or two capitals, you will show that trait. Recessive features, these are the ones that we represent with a lowercase letter on our diagrams, and the only way you will see that phenotype is if you've got both recessive alleles present. When we then come on to try to work out what parents will have just from a written question, they're going to use the words homozygous and heterozygous. So homozygous, homo, same, zygous is referring to the alleles. So if it's homozygous, they're going to be the same alleles. So they'll either both be capitals or both be lowercase. You know which one it is because the second word after the word homozygous will be either dominant or recessive. So if it says homozygous dominant, both capitals. If it says homozygous recessive, both lowercase. Heterozygous, break it down again. Hetero means different, zygous, alleles. So you would have one capital and one lowercase. So you can work out whatever alleles are present within the parents from those terms. Now, this is our little Punnett square, okay? It's basically like a slightly extended window frame. Now, these are really straightforward to do. So I've already put these ones on here just for speed today. But when we come to fill it out, whatever is sitting at the top here, you literally put in the two boxes beneath. So all we do is put our little alleles in the two boxes beneath. Then we do the same, start at the beginning of the row and put it in the two boxes to the right. So all we're going to do is fill those in and you've got your potential offspring. Now, what we then need to do is obviously give a percentage or a ratio or a fraction or a decimal relating to how many of them would show a certain trait. So in this case, if we were looking for a recessive trait, then we would only have one potential, which would be 25%, because there's only one that has both lowercase letters. That would be the only one that could show the recessive feature. If it asks you for the dominant feature, then it would be 75%, because as soon as they've got a capital in there, that's the feature we show. The only other one that they can ask you to do a Punnett square for would be the inheritance of sex. So hopefully we remember that males have one X and one Y chromosome, and females two X chromosomes. Now, the good news is this never changes, because obviously for sexual reproduction, you need one male, one female. So we'd set out a little Punnett square like this. Same thing, whatever's at the top goes into the two boxes beneath, whatever's on the left goes in the two boxes to the right. So when you actually fill it out, you will end up with 50% chance of male and 50% chance of female each and every time. If they do ask you to write down the chromosomes present in a male or a female, please don't just write X or Y on their own. You need both of them. So male must say XY, female must be the two X's. Don't just write X because they're both X's. XX. The next thing we need to know is mutation. Now, when we're talking about mutations, these are changes in the base sequence of the DNA. Now, these can occur spontaneously, but their chance will increase if you are exposed to things like ionizing radiation, like UV, or certain chemicals. Now, what we actually find is that these mutations can lead to variation, but a lot of them will not really change the phenotype. So even if you do have a mutation, there's a number that just show no effect on the phenotype at all. So we won't actually see any difference. Now, when we do get a mutation, they could be harmful, think cancer. They could be beneficial, the ability to speak, or they could be neutral, your ability to roll your tongue or not. So different mutations have different benefits or drawbacks. If you are doing GCSE biology higher tier, this bit is for you. The rest of you don't need to know this. Now, when we're looking at DNA, basically a strand of DNA is split up into these coding and non-coding regions. So you have a little section of DNA that codes for something, and then it's split off from the next coding section by a bit that's called non-coding DNA. So when we get a mutation occurring, if it happens in the non-coding region, it has zero effect because that bit of the DNA doesn't code for any proteins at all. It's kind of like a spacer bit of DNA, I suppose. If the mutation occurs in a coding region, then obviously that can lead to some changes in our organism. Now, the way that we can see these changes, there are three key things to remember here. We can do a simple substitution, so swapping one base for another. We can add a base in, or we can delete a base. So if we've got our little strand of DNA here, going back to our module 1 stuff again, we're looking at protein synthesis. Here's our DNA, and remember, each little sequence of 3 bases is going to be coding for an amino acid. Now, if what we're doing is just swapping a single base, so if we change this first C, let's change that T here, in fact. So if we were to change this T for an A, then we would change that first amino acid. All the others would be the same, because obviously when it's read in that little triplet, we'd only change this one. The next thing is that if we were to have an addition, so if we added a base in here, what's going to happen is we're then going to have another base in this point, which means it will be this one and these two making up the next triplet. It's got the knock-on effect on all of the others after that point. Same idea with a deletion. If we take this one out, for example, then we're going to be reading it in a different frame. So in each of those cases of the deletion and the addition, big effects, because it affects every triplet that comes after that. Now, what we do find here is they can link this back really nicely into that B1 work that you've done on transcription, translation, and potentially the enzymes. Because if we're changing, obviously, the amino acids present in an enzyme, which is just a protein, we're changing the shape of the active site, and therefore it's no longer going to be complementary to the substrate. So remember to look out for some of those links. Still just higher tier biologists, you need to know about good old Gregor Mendel. So this was the bloke that sat around in a garden playing with pea plants. What he then came up with was this idea that we are going to get characteristics passed on from from their parents to their offspring. Now, what we're going to see here is he's got this whole idea of these hereditary units. So something being passed from parents to offspring that will either be dominant or recessive. So he kind of came up with this idea of things being passed on. The next scientist you should be aware of is a guy called Misha, and he basically discovered there was this acidic substance in the nucleus. Avery was the one that then transferred some DNA between bacteria, and what he saw was that you could actually transfer an ability from one bacteria to another when you do that. So he showed that genes were made of DNA. Chargaff is probably the next one that I would really concern yourself with here, other than Mendel, because Chargaff was the one that discovered those base pairs. So look out for these questions where they're going to tell you something like 20% of the bases were adenine, and then ask you what percentage of the bases are guanine. So if we know 20% are adenine, 20% must be thymine because they're complementary base pairs. Leaves us with 60% of our DNA now. And again, that'll be divided by 2 to give us 30% for the other one. So just look out for that simple maths, remember your base pairs, and you can calculate. The next ones were Rosalind Franklin and Morris Wilkins. Now, Rosalind Franklin was the lady who did the X-ray photograph of the DNA crystals that Watson and Crick then used to come up with the double helix structure of DNA. So again, link back to our earlier work on DNA and its actual structure at that point. So what we're going to see then is modern times, we're now looking at how we can use this information that we've now got all about DNA for our benefit. So we did this thing called the Human Genome Project back in the early 2000s, where basically we sequenced all of the genes in the human body. And we're going to look and see why that was important towards the end of Unit 6. But the key thing is that when we sequenced that genome, we learned about where potential problem genes could be, and therefore potential treatments for them as well. So back for everyone now. So triple, combined, foundation and higher folks, we need to know about evolution. So evolution, quite simply, is the gradual change in a species over time. And the way that organisms evolve is through the process of natural selection. If you are asked a question on natural selection, they're usually really good, because they usually sort of ask you something and then they say, use ideas about natural selection in your answer. So talk about these key things. First one, there is variation within a species, usually caused by mutation. Step 2, those that are best adapted to their environment are more likely to survive and reproduce, which means they are going to pass on the advantageous alleles to their offspring, and it repeats over many generations. 5 key things, okay? So variation within a species caused by mutation, best adapted, more likely to survive, reproduce, pass on the advantageous allele, and repeat over many generations. I will answer questions at the end, so stick with this at the moment. We do have a couple of examples of this actual evolution taking place in natural selection. Think about the peppered moths, different colours. Ones that were camouflaged, weren't eaten, their population increased. The ones that weren't camouflaged, got eaten, they decreased in number. We also have antibiotic resistance in our bacteria. Those that are resistant to antibiotics are more likely to survive and pass on that allele to future generations. In terms of providing evidence for evolution, then what we're going to find is the fossil record is key here. We can collect all these fossils of organisms that used to live, put them all together in chronological order, and then look at these teeny tiny little changes that have taken place over all these thousands or millions of years. This then supports this idea of small changes over a long period of time. We can also see some more modern evidence by looking at DNA and proteins and comparing them. The more similar organisms' DNA and protein structures are to one another, then the more closely related they will be. So what we can see there is if we compare, say, our DNA with a chimpanzee, there are very minor differences. Therefore, we're quite closely related. Hopefully an obvious point that if a species doesn't actually manage to adapt to any changes in their environment, so they're not able to cope with a change, then they are going to become extinct. So we've got endangered, risk of extinction, extinct, they're all dead. Right, GCSE biology folks only, foundation and higher, you need to know about good old Charles Darwin. So hopefully we do know that Charles Darwin was the guy that went and sailed around the Galapagos Islands on the HMS Beagle. And when he was doing that, he was literally having a little look at all these different things that lived on those unique islands. The key ones he looked at were, of course, the finches, the little birds. And he noticed that they had different beaks depending on the food sources on those individual little islands. And that was what he based his theory of natural selection by evolution on, okay? Secondly, we have another scientist at the same time as Darwin called Wallace. No one cares about first names, by the way. Darwin, Wallace, all good. Now, Wallace was working basically on the other side of the world at the same time that Darwin was sailing around the Galapagos Islands, doing his own studies, looking at his own organisms, and he actually came up with the same idea as Darwin. So 2 scientists working completely independently, but they came up with that same idea. Darwin is the one that's more known because he went on to publish a book that made his theories accessible to the general public. Whereas previously, if you only publish your work in scientific papers and scientific journals, that goes to scientists. So obviously useful in the terms of peer review, people can read it, check your work, obviously advance the work, etc. but it won't get to the general public so easily. So because he published a book, that did go to the wider public, and therefore we gained greater knowledge of Charles Darwin. Last little bit of B5 is for combined and triple, folks, and this is all about classification. So classifying is pretty much sorting living things into groups based on their characteristics. Think of it as a massive filing system for living things. Now, why do we classify? Number one, makes it easy to identify species, and we can show these links between them. When we are talking about this term species, we need to know that a species is a group of organisms that are able to interbreed, so breed together, and produce fertile offspring. So 2 key points there: group of organisms that can breed together, secondly, produces fertile offspring. When it comes to our classification, we have got 7 taxonomic levels. You need to know the sequence of these. So I always suggest making up your own little rhyme to help you remember it. So we've got the kingdom, the largest of them. Kingdom, phylum, class, order, family, genus, species. You need to know that sequence because they can give you a little table with some bits missing. You might need to fill it in. 5 kingdoms that we classify all living things on planet Earth into: plants, animals, fungi, prokaryotes, and protoctistans. Now, in terms of how we're going to sort these things into those groups, we have got 2 potential systems. First one, artificial classification, purely looking at observable features. Does it have wings, etc.? If it has wings, we chuck it in a group together. It doesn't consider any of these evolutionary relationships and links, though. The second one is this natural classification system, and this is all based on DNA analysis and seeing how similar the DNA is. So this leads on to this whole idea of phylogeny, which is where we can create those phylogenetic trees that show how closely related organisms are. So the phylogenetic trees, you've got these little branches, and they all connect going further and further back in time. So the more closely related they are, then the closer those little branches are on that phylogenetic tree. Right, that's B5. Last one, B6, which is a long one. So B6 is pretty much like 3 different chunks of biology. First chunk is all about sampling and biodiversity. So sampling techniques, there's a whole range of different ones that you may have encountered, things like pitfall traps, pooters, sweep nets, etc. But the 2 that are more likely to come up are going to be our quadrats, so those fixed area squares, little grids, and a transect line. Now, when we're talking about a quadrat, what we are going to do is obviously use it to take samples from a field, for example, of plants, okay? Now, when we are doing this, first thing is we need to decide if we're going to do random sampling or a systematic sampling. If we want to just get a population estimate of plants in a field, we would do random. The way we would do that, 2 tape measures at 90 degrees to each other, that creates a nice grid in our field. Use a random number generator to create some coordinates, and then you take your quadrat and place it at those coordinates, counting the plants in each one. You then do a certain number of quadrats, we know the total area of those, multiply it up to the total area of the field, and that gives us our population estimate. Reason that we do that is it avoids bias. If I was to sort of say to you, right, I want you to go out and then give me a population estimate of all the plants on the school field, after you counted the 78th square of just grass, you start looking for the dandelion, the daisy, etc., because it's boring, and therefore that's going to lead to a false population estimate. So random avoids that bias. If, however, we wanted to do a sampling to see how something like light intensity affected a population, we run a tape measure or a long bit of rope out from under a tree into an open area. Then we'd place our quadrat at fixed intervals and count the plants present within each of them. That then allows us also to measure the light intensity at each of those points, and from that we then have our nice little graph that shows us how the populations and distributions change with a particular abiotic factor. So that's a transect line where we're running this line out across a certain abiotic factor using the quadrats then to get a count of the different plants present at each point. One thing that we need to be able to do is identify stuff that we find, and this we use a key for. There are two types to know about. The first one is a spider key, which is literally where you've got those little arms coming down. So you have something that says basically the leaves are going to be broad or narrow. So you just go whichever branch is accurate for what you're looking at. Keep following those little lines down until you hit an organism at the end. Option 2 are the numbered keys, which is kind of like one of those pick-your-old adventure books that you might have read as a kid. So what we do there is we read the question, and then it tells you, go to question whatever number it is. Just follow it through again. Eventually, it tells you the name of an organism. So all we're doing there is we're using observable features to give us a name. They can give you little pictures of organisms and a key and ask you then to work out what they are. So make sure you can do that. Another maths one is the good old capture-recapture technique. So capture-recapture is something that we'd use for things like, I don't know, grasshoppers. If you wanted to know how many grasshoppers there were in a field, you go out with your big sweep net, you do a collection of grasshoppers, we'd count how many there were, and we'd mark them in a subtle way. The subtle way means we're not going to kill them through the marking or make them more likely to die as a result of it. So you wouldn't put a bright pink bit of nail polish on their back because you know what? Birds can see that. So we got to mark them in a way that means they're not likely to be killed more than any others. Then we release them. At a later stage, we go back out, do exactly the same sampling technique. We count how many organisms we've caught and how many of them were marked. From that, we can then calculate the population size. So total in your first times total in the second sample divided by the marked ones. That gives us our estimate of the population size. It does make two big assumptions though. Number one, nothing died between your first and your second sample. And secondly, nothing either entered the area or left the area. So no immigration or emigration of that particular species. The next bit we're going to look at is biodiversity. So biodiversity is just the variety of living things we find in a given area. And what we're going to find is humans basically create a problem for biodiversity. We are the leading loss of biodiversity cause. So first thing we do, deforestation. Cut down huge areas of forest, massive amounts every single day. Reducing trees reduces habitats, reduces food supplies, etc. Second one, we've got a lot of farming because we need to meet the food demands of our growing population. Therefore, we're changing all this diverse land into basically a giant field of just the one plant. The calculation for capture-recapture is quite simply the total that you caught in the first sample times the total you caught in the second sample divided by the ones that you re-caught that were marked. So the second sample marked ones. Next thing we've got is the hunting and fishing aspect. So obviously we go out catch fish, sometimes for food, sometimes for fun. And of course hunting, sometimes for food, sometimes because people just like to kill things. So obviously that's going to affect the number of organisms that are existing, could affect food chains. And then our probably biggest issue, I would say, is pollution. So more that we pollute our planet, the fewer organisms that will be able to survive there. Some are way more susceptible than others. One of the big problems we have is coming from basically our farming techniques where we are using all these fertilizers on fields because we get this process called eutrophication. Fertilizers will wash off the fields, end up in a water system. Because they've then got loads of these nutrients, algae is going to have a population explosion, something we call an algal bloom. That's then going to create a block to any light going into the water. Any of the plants that were living in there will die because they don't get the light for photosynthesis. They will decompose. As they're decomposing, then we're using up the oxygen in the water, and that is going to kill the fish. So quite a detrimental problem in the grand scheme. We've already mentioned this difference between endangered and extinct. So endangered, at risk of extinction, and extinction, all dead, all gone. What we're trying to do to combat this is coming up with these ideas of conservation techniques. So conservation is trying to protect the environment to ensure we're not losing habitats and therefore not losing species. And one thing that we're trying to do is basically use resources in what we call a sustainable way. So this is basically using resources today, but making sure that they're still going to be there for future generations. Now, ways that we can do this is we can do things like restricting human access. So if we obviously know that there's particular species that breeds in one area at a certain time of the year, stop humans parading around in that area at that point of the year so those animals can breed happily. If we're looking at some of the kind of more invasive ways that we can help, then we could do captive breeding programs that then lead to reintroduction of species where we've reduced their numbers in the wild. Now, captive breeding is where we are using, obviously, populations within zoos, basically, to breed together to increase the numbers. The idea being that we will then reintroduce those to their natural habitat. However, there are problems. First one is if we've only got a limited number of these organisms within our captive breeding population, they've got a limited gene pool. So when we're just breeding them together over and over again, we're reducing the size of that gene pool, and therefore we can actually see more problems in terms of recessive diseases coming through. Secondly, if we've bred some of these organisms in captivity, they just may not be suitable for release. They may not have those survival instincts where we fed them forever. They may not know that, you know what, that creature over there is going to kill you if you go near it, because they've never seen it before. They haven't had that kind of training through growing up in the wild. Or there just may not be a habitat to go back to. If where they used to live is now a huge city, you can't very well just release them again. We don't just do it with animals, though. We also do this with our plants through the use of things called seed banks. Now, a seed bank is basically just a massive store of seeds of a huge number of plants. Idea being, something catastrophic happens, we can just go to the seed bank, get the seeds out, and then grow a new population again. That's the theory, anyway. We do also have a lot of work going on legal grounds here, because to conserve this biodiversity, yes, we need to work in our local area, but it can't just rely on a small community doing this. It's going to have to also involve wider populations, so potentially international cooperation here. Therefore, we do have a couple of key things to know about. We've got the IUCN Red List, which is basically a list of species that need protection, and that's updated annually. And then we also have CITES, the Convention on International Trade in Endangered Species of Flora and Fauna. So basically, it's a whole load of regulations on what you're allowed to import and export around the world, and not only as a whole organism, but as products. So that's the bit that's banned things like trading ivory from elephant tusks, etc. So we do have some of these international agreements all geared towards obviously conservation, reducing the impact on our living things. Now, one other thing to be aware of is this thing called ecotourism. And tourists, bit of a double-edged sword. One thing that they are good at is bringing money to an area. So if we can find a way to encourage tourists to come to the area to see these creatures in the wild, brilliant. They're going to bring their money with them. That money can then be used for conservation efforts in the area. But we do need to be mindful of the negative impacts as well. When we're bringing more tourists in, then obviously there's a greater risk of rubbish. There's a greater risk of them trampling on stuff if they don't follow the rules of stick to a path, etc. So there are some drawbacks to this ecotourism. But main idea being, if it's done well, we can bring money in, which we can then use for further conservation techniques. Higher biology folks only, you need to know about how we can monitor biodiversity. So what we see here is, to monitor it, we need to take samples. So we need to have that regular check on how many of those organisms are actually present. Another way we can do this is by looking for something we call an indicator species. Now, indicator species are clever little things, because basically whether those species are present or absent tells us something about that environment. So it might tell us whether the water is polluted or whether the air is polluted, just based on what is actually alive in that particular area. So still just higher biology, folks, you need to know about air pollution and water pollution. Air pollution, first of all, most common problem, sulfur dioxide from all these internal combustion engines on our cars, etc. When we're burning all these fossil fuels, we're going to be releasing sulfur dioxide into the atmosphere. As that dissolves in the clouds, we're going to generate acid rain. Now, acid rain is problematic because if it falls on things like our trees, it can kill the little meristems at the top where it's growing. And if it builds up in water systems like ponds and lakes and the like, change the pH and can kill the fish there as well. So quite damaging in its very nature. How can we use an indicator species to work out air pollution? There's something called a lichen. Now, I'm sure if you look outside of any near window, a rooftop nearby, you can probably see something kind of either white or yellow sitting on that roof. That is going to be a lichen. And usually that's one that grows where there's lots of high nitrogen content, because that's where the birds are pooping, and that's got a lot of nitrogen. Lichens are great for indicating air pollution though, because they don't have a root system, so all of their nutrition just comes from the air. So if we've got a polluted air sort of area, then what we're going to find is we're not going to have our full range of lichens. We're only going to have those pretty tough ones that are good at growing in high levels of pollution. In terms of water pollution then, this is going to be looking to see what organisms are present within the water. As we've probably seen on the news then, the water companies are in a lot of trouble around the country because they keep releasing raw sewage into our water. That's problematic. And therefore, what we're going to see is the amount of pollution in that water is going to affect what can live there. Main reason, higher pollution reduces the amount of dissolved oxygen. So a few key species we can know about here. If you've got these little things called mayfly larvae, they will only live in beautiful crystal clear water. So if mayfly larvae are present, you've got no pollution problem whatsoever. If, however, you have delightful creatures called things like sludge worms, rat-tailed maggots and the like, then you've got a pollution problem there. Generally, if their name sounds a little bit unpleasant, it generally means there's lots of pollution in the water. And yeah, they are crusty lichens, the ones that are sitting in those high pollution areas, generally flat, crusty, very unpleasant looking. Low air pollution areas, they're all lovely and branched and very frilly looking things. Right, back for foundation and higher, but only the triple biologists at this time. We're on to our next section, which is all about basically feeding people. We need to know the word food security. So food security is basically our ability to meet the demands of the human population by providing everyone with enough affordable food of sufficient quality and quantity. So the food has to be the right amount and the right quality for everyone in the population. There are a few key threats to our food security. First one, the ever-increasing population of us as humans. That is a problem. Secondly, we are going to see the issues of climate change, meaning not everywhere is suitable for growing food and therefore putting further restrictions on how much food we can produce. And then we could also see things like pests and pathogens evolving. How can we get around this? Some of our traditional techniques, we can obviously use things to maximize photosynthesis for our plants. Stick it in a greenhouse, okay? So basically, if we're growing our food in these massive greenhouses, then we can control the temperature, the light intensity, etc. Whole lot, maximize photosynthesis, maximize the yield. We can use our chemicals, so fertilizers, pesticides, insecticides, herbicides, all of those are going to help us to maximize our food production as well. When we are talking about those chemicals, remember, side means kill. The thing in front is what it kills. So fungicides kill fungi, insecticides kill insects, and pesticides kill pests. The only one not to use that simple rule with is herbicides. Herbicides don't kill herbs, they kill weeds, folks. Okay, so just remember that last one and don't make a fool out of yourself. It's not like we're spraying chemicals just to kill a bunch of oregano in a field. We then have two options available to us for food production: intensive or organic farming. Intensive farming is basically where we're going to maximize the food production in a given area. We're going to use chemicals, we're going to use machinery, anything at our disposal, we're going to throw at this to get maximum yields from that area. Organic farming, kind of the opposite. What we're looking at there are the more natural methods of producing crops and animals, avoiding those chemicals. So rather than using a chemical fertilizer, we'll spread manure because it's natural. Rather than spraying our entire crop with a chemical herbicide, we're going to go out and pull the weeds up by hand. So obviously, different techniques, different pros, different cons there. So one thing that we should know is this phrase, biological control. When we are talking about biological control, we are using a natural predator to eat the pests. So things like a ladybird to eat aphids. There are some downsides to this because obviously if you are trying to use a natural predator to cope with a pest problem, then they are living things. So when we release those little predators, they may just go somewhere else. They're not going to eat the pests in your area then. Or in that field or greenhouse, they may see something that they prefer the look of and the taste of and eat that instead of the one that was creating the problem for you. So biological control, natural predators to eat the pests, but it's not perfect. So other techniques that we can use that are more modern then. Fish farming is the first one, where basically we have these giant cages in the seas. We put fish in those cages, they breed, we can then catch them straight from there. This means that we're not going to have to go out and fish wild populations, gives them a chance to recover, and it means we can catch fish whenever we want. We're not going out on fishing vessels hoping we find a shoal of fish. We're literally going to the cage, drop the net, there they are, job done. Downsides to it, obviously, we've got fish in a cage, they're all close proximity. That means diseases will spread quite readily. And it's a cage. That means the wild fish can swim right the way up to the bars, and therefore diseases could spread to those wild populations as well. Next technique is one called hydroponics, and this is where we are able to grow plants not in soil. So we're growing them basically in a solution that contains all of those minerals that they need to grow. Now, big advantage of hydroponics is because they don't need soil, we can stack them up. And you can also grow them wherever you want. Poor soil quality, a shipping container, underground, you name it, we can grow plants there because the soil is irrelevant, we don't have it anymore. If you can put the solution in there and you can put a light in there, you can grow plants there. Right, back for everyone, triple combined higher and foundation. We need to know about selective breeding next. Selective breeding is a simple process where we can maximize a certain feature. We look at our particular organisms, whatever they may be, we decide what feature we want, and we then select the organisms that have that characteristic. So we then breed them together, we then collect the offspring with the best characteristic again, breed them together, and repeat over many generations. So if you get a question asking about how we carry out selective breeding, identify the desired characteristic, breed the organisms with the highest quality there, and then collect the offspring, breed them, repeat over many generations. Downside though, we are just breeding a small population over and over again, which does mean that we are reducing our gene pool, making them more susceptible to potential environmental changes and more likely to inherit recessive genetic diseases as well. The more modern technique of this is genetic engineering, where we are going to alter the genome of an organism by selecting genes for our desired characteristic and basically take it from one organism and stick it in the other. Now, what we will see here is that what we're going to actually be able to do is not only focus on one species, we can take a gene from a completely different species and stick it into something else. So we've done things like taking a gene that creates a certain toxin from a bacteria, putting it into a corn plant, makes it resistant to a pest. So there's a lot more flexibility there, and it's much quicker when we're doing that. GCSE biologists, you need to know the downsides. Key one, we don't know the long-term effects of this stuff. We've not had it around for that long, so long-term effects are still unknown. Possible negative effects we could see, some health problems, because we are introducing new genes into an organism. So you might create an allergy that was unintended. And we could see cross-pollination with wild plants, leading to unforeseen problems as well, disrupting food chains, etc. Anyone doing higher tier on either combined or on triple, you need to know how we do this in a teeny bit more detail. So there are going to be two enzymes you need to know the names of here. First thing we do, once we've identified the gene that we want to cut, we use this thing called a restriction enzyme to cut it. So restriction enzymes cut the gene, and we're going to cut a plasmid, that little loop of DNA in our bacterial cell, with the same restriction enzyme. We then put those two things together and we join them with a second enzyme called DNA ligase. So ligase joins those two things together. We then put the plasmid back inside the bacteria. We allow it to grow in large numbers because remember, bacteria reproduce asexually, so they will all be genetically identical. And we're going to do this in a huge vat, basically, and then we can just collect that product. Best example, human insulin. Took the gene for insulin from a human cell, inserted it into a bacterial cell. We now have bacteria that make human insulin for us. One other thing we should know about is we need to check to make sure that gene has been taken up, and the way that we do this is with a marker gene. Two options available. First one is antibiotic resistance, where we will put a gene for antibiotic resistance alongside the desired one. Second one is a fluorescence gene. So basically the idea being that if the organism takes up the desired gene, it's taking up that other one as well. So with antibiotic resistance, just grow it on a plate that has the antibiotic present. Only the ones with the resistance gene will grow. In theory, they've got the one you wanted. Or we can do the fluorescence, grow it on a petri dish, shine a little light on it, and then they will glow if they have got our desired gene present. Back for foundation and higher, combined and triple, folks, we need to know about biotechnology. When we're talking biotechnology, this is basically using a biological process or a living organism to make a product. I don't do predictions. It's a fool's game. Just learn the stuff. Now, in terms of what we're going to produce, these genetically modified organisms. Now, these are going to be organisms that provide an extra bonus to us. Good example is golden rice, which is typical rice, but we've inserted a gene from a daffodil into it. Whole idea behind that is that that rice is then going to produce this stuff called beta carotene, which is going to be used in the body to make vitamin A. And vitamin A is necessary to avoid blindness. So by producing this golden rice, we've then been able to stave off this whole idea of blindness in huge numbers of people. For those of you doing higher tier, just be aware that if we are transferring foreign DNA from one organism to another, we use this thing called a vector. And that vector could be a virus or a plasmid most commonly. Okay, last section, the disease bit of Module 6. And this is for everyone, combined, triple, foundation, and higher, folks. When we talk disease, we are talking about two categories we can split it up into. Communicable, ones that spread from an organism to another. Non-communicable, don't spread between organisms. If it's caused by a microorganism, then we call that a pathogen. So pathogens are microorganisms that cause disease. You should know some examples of both plant and animal diseases caused by a fungus, a bacteria, a virus, and a protozoan. So the fungal diseases, athlete's foot, that's the one for our animals. That's your kind of itchy, crusty flakiness between your toes. We then have powdery mildew in our plants. Powder sits on the leaves, reducing the amount of photosynthesis. Bacterial disease in animals is tuberculosis. Basically, the bacteria eats away at the lung tissue until eventually it is going to rupture blood vessels, leading to you drowning in your own blood. Delightful. And we've got crown gall disease in our plants, where basically a gall is like a swelling around the actual plant itself, impeding transport. Viral disease, take your pick. Influenza is a good one though. So influenza in our animals, the flu. And then in plants, we can have tobacco mosaic virus. Changes the leaves, discolors them, mottles them, generally reducing photosynthesis again. Protozoal disease is the last one. Malaria is the best example for animals. And obviously, malaria is a disease that is going to be spread by a vector, the mosquito. And in our plants, we've got coffee phloem necrosis. And anything that's necrosis is basically breaking down of tissues. So, non-communicable diseases, not spread by pathogens because they are not spreading, okay? These ones are going to be caused by issues with the body usually. So it could be that your diet is pretty terrible and therefore you're not getting enough vitamin C, so you're going to get scurvy. It might be that you are obese and therefore more likely to develop cardiovascular disease or diabetes. You could have a genetic disorder, cystic fibrosis, Huntington's disease, for example, or you could have a body process not working properly, like no control over your cell division, leading to cancer. What we do find is that there are some of these that are actually linked. So they've got separate causes, but they're linked conditions. The one you're most likely to know is HPV. So HPV is a virus, and that's obviously going to spread from one person to another. But HPV is linked into developing cervical cancer. Obviously, cancer is not communicable, but HPV, which is linked into causing it, is. So we've got those two little links there. The next little section is how we spread these communicable diseases and obviously how we can avoid the spread. So what we then have is spreading communicable diseases. Think about ways it's going to get into the body, breathing it in. Okay, you could have sexual contact. We could be talking consuming it in food or drink, cuts. All of those are going to potentially lead to infection from a pathogen. One phrase to know here is droplet infection, which is basically if you've got terrible manners and you cough or sneeze without covering your mouth and nose, then what we're going to see is you are going to basically end up with this cloud of little droplets filled with pathogens splattering the faces of others around you, surfaces, and potentially others inhaling it. Now, obviously, if that happens, you're likely to obviously get that illness as well. Now, we don't, the moment someone coughs on us, develop the illness. There is a lovely delay called the incubation period, which basically is that time between infection and actually developing those symptoms. And the reason we've got that incubation period is those pathogens have to replicate in our body. Bacteria do this in a process called binary fission. So they're dividing by mitosis, doubling their population each time. And that gives us this exponential growth rate. Viruses are going to do it slightly differently. They actually insert their DNA into the host cell, basically take over the host cell and use it to make more virus particles, which will then just erupt from that host cell to then infect many more with all those new viruses it's just created. The next thing is, how do we stop getting infected by these communicable diseases? Now, I'm not being funny, but if you can't actually give me a couple of marks on this after living through a global pandemic, then I am seriously concerned. Think everything you did in COVID, washing hands, wear a mask, social distancing. We can then go to other diseases as well, spread through other means. If we're talking about sexually transmitted ones, use a condom, avoid having sex. If it's something spread through other body fluids like blood, don't share needles, obviously. All of these are different ways that we can avoid spreading diseases. Now, what we then need to think about is going to be our plant defenses for those of you doing the biology. So higher end foundation, GCSE biology folks, we need to know about plants and how they're going to defend themselves against these diseases. Two ways: physical defenses, and basically you need to know cell wall and you need to know waxy cuticle. So the waxy cuticle, that layer of wax on the surface, meaning water doesn't sit there, so the fungal spores won't be able to germinate there. And it also just prevents other things getting through to the more delicate cells underneath. And the cell wall, a physical barrier that makes it harder for pathogens to gain entry to the cell. Option two, the chemical defenses. Our plant will secrete certain chemical substances to kill a particular microorganism. So obviously, if it's an insecticide, it kills insects. If it's an antifungal, it will kill a fungus, etc. For those of you doing higher biology, you need to know how we can actually identify these different plant diseases. So what we're going to see is we've got field techniques and we've got lab-based techniques. In terms of the field techniques, two options: observation. What colour are the leaves? What's happening to the leaves? Are things rotting on it? All of those. Basic observation. Certain things will lead to a certain disease diagnosis. Or microscopy. We can take a light microscope out into the field with us. Remember, they're small, they're portable, they're cheap. That means that we can literally take it out, create a little slide, have a look. That might allow us to identify what pathogen is causing our problem. Downside, we need this plant to be showing a sign of a disease for us to be able to identify those problems. Option 2, the lab techniques. This one will use DNA analysis. So we'll take samples from our actual unknown pathogen, run it through a gel electrophoresis, and then we can match that banding pattern to known pathogens. Or we could use antigen identification, where basically antigens, those little protein spikes on the surface of the microbe, and we can then carry out a chemical test to identify which ones are present. Back for everyone, combined, triple, higher, and foundation, we're looking at how humans defend themselves against the disease. First thing we need to know, blood clotting. If you cut yourself, you will form a blood clot, hopefully. Reason for that is that you will then not bleed to death. So how does it happen? In our blood, we had those 4 things. Hopefully you remember them. We have red blood cells, white blood cells, plasma, and platelets. We're looking at the platelets at this point. So when you cut yourself, the platelets are going to go to that region, and they're then going to cause this change from a substance called fibrinogen into fibrin. That forms like a mesh, like a little net, and that's then going to trap red blood cells within this little mesh network, and that creates a blood clot. As they then dry out, that is a scab. So that scab is then going to sit over the wound, preventing pathogens gaining entry, preventing you bleeding to death, until your body has regenerated those skin cells using mitosis. We then come on to the non-specific defense mechanisms, which are basically not going to be geared towards any particular microbe. It's a one-size-fits-all approach here. So this is things like your skin being a physical barrier, just stops things getting into those more delicate cells underneath. You've got your acid in the stomach, which will kill microbes on anything you're consuming in that way. You will have things like your tears on your eyes, which contain enzymes to break down any pathogens that may end up on the surface of your eye. We could go into the airways, where we've got the mucus, traps the pathogens, and the cilia, those little hairs that do their little wafty motion, move it up to the top of the throat, where you can then swallow it down. When we move on to the more specific response, then we have 2 types of white blood cell we need to know about. Phagocytes engulf and digest the pathogen. Don't say it eats it. It engulfs and it digests. That is the process of phagocytosis. Option 2 of our white blood cells are the lymphocytes, and these are the ones that produce antibodies or antitoxins. Now, they can give you a little blood smear, sort of microscope image of a blood smear, and ask you to identify things. Red blood cells are always the little pink-looking things. Your white blood cells are going to be the purpley-looking ones, larger purple blobs. Now, obviously, you've got 2 types. Your lymphocyte is the one with the huge nucleus basically filling the whole thing. Your phagocytes are the ones where the nucleus looks like it's got little lumps sticking out. So you should be able to identify those from a blood smear. We then need to know what on earth an antibody is and what it does. Antibodies are just proteins that are going to bind to the antigens on the surface of a pathogen. So what we then find is once the antibody that's released by our lymphocytes has bound onto the antigen on the pathogen, then our phagocyte can engulf and digest it. Because this antibody is specific to one antigen, then it means this is a specific form of defense. The actual antibody is complementary to just that one antigen. So obviously it's specific to that one pathogen. The good news is when you've been infected by a pathogen, your body has created these antibodies, then you are going to keep some of the cells that make those antibodies as memory cells. and they're going to remain circulating your body for a period of time. Okay, depending on what type of cell it is determines how long it stays. Some short, some much longer term. The whole idea behind that and these little memory cells is that if you are then exposed to that same pathogen a second time, you are going to be able to mount a much faster response. Now, normally that's quick enough to stop you showing any symptoms at all, and that is our immunity. For those of you doing higher tier biology, you need to know about these things called monoclonal antibodies. Now, a monoclonal antibody is something we make in a lab using a special type of cell called a hybridoma. Now, hybridomas are made of two cells. We make them from a lymphocyte and a myeloma. Myeloma is a cancer cell. Lymphocyte is obviously a white blood cell that makes an antibody. We fuse those two together to make a hybridoma, hybrid of two types of cells there. Now, each of these hybridomas is going to be able to produce a specific antibody, the monoclonal antibody. And the idea being that those monoclonal antibodies that we are making will bind to a specific antigen for a certain purpose. They could ask you a little bit of detail about how we do this in the lab. So we start off with a genetically modified mouse and we inject it with the required antigen. That mouse is then going to obviously make antibodies to the antigen. So it's going to make those lymphocytes that create those antibodies. We can collect those lymphocytes, fuse them with the myeloma, the cancer cells, to make our hybridoma, and then we will be able to have those cells reproducing to form all of those antibodies that we need. The uses for the monoclonal antibodies, most common one is pregnancy testing. So those little pregnancy test sticks that you can just pee on and get your little positive, that uses a little HCG receptor basically. So HCG is something that's produced once you've got a baby that is developing. Now, we use these little monoclonal antibodies that are stuck basically on a little pad in that little test stick. And if the little HCG is present, it's then going to basically move a little bit of dye, stick onto a line, and you get a little plus or a little line showing. We can also use these monoclonal antibodies for detecting certain diseases, because again, if a disease is present, then we can get that little quick rapid test. I think COVID tests, you all did those, I'm sure. And we may be able to use them in terms of treating certain types of cancer. So using a monoclonal antibody that will target a specific cell because it's going to be complementary to a receptor on its surface, hopefully delivering a drug to reduce side effects of those cancer treatments. Right, back for combined triple, higher and foundation, folks. We need to know vaccination next. When we talk vaccination, this is something we will deliver as an injection, and it will contain either dead or attenuated, which is weakened versions of the pathogen. That means our lymphocytes are going to detect the antigen, create the immune response, but we are not going to get sick because this pathogen is either dead or it's weakened. It won't cause disease. So we're getting the benefit without the downside of getting sick. Because some of those lymphocytes will remain as memory cells, we have immunity to that particular infection. We then have a few very similar terms that we need to know the meanings of. Antiseptics, first of all. Antiseptics are chemicals we will use on external living surfaces, like your skin, to kill a pathogen. Now, the good news about these antiseptics, they'll kill a pathogen, won't harm your cells. So think about alcohol. So when we're using all those hand gels, that was using alcohol to obviously kill any of those pathogens on our hands, but the skin was absolutely fine. So antiseptics kill living things on living surfaces. Disinfectants are going to do that same job, but on a non-living surface. So when you were wiping down desks in COVID, that was using a disinfectant, killed the pathogen on a non-living surface, the desk. We then have our antivirals. Antivirals are basically drugs that kill viruses. It's in the name. Now, what we generally find is that these will act on just a single type of virus or a limited number of viruses, and their side effects can be a little bit unpleasant, but they work in a range of ways. Key thing, though, is they're going to try to stop that virus from replicating, possibly by blocking it getting into the host cell, possibly by stopping it from getting its genetic material in there. It's going to stop that replication process somehow. The other drug we have are the antibiotics, and these are the ones that kill bacteria. So look out for those questions that ask why we wouldn't treat an influenza infection with an antibiotic, because influenza is caused by a virus and antibiotics only kill bacteria. And again, they work in different ways to kill the bacteria that's present. This ties into one of those practicals that hopefully you did in class, where you would have had a little agar plate and you then would have had little discs basically with different antibiotics on there. That agar plate would have been seeded with bacteria. You then put those little paper discs on the surface, incubated it for a little while, and then had a little look at these clear zones, the zones of inhibition. Basically, a zone of inhibition is a region where our bacteria cannot grow due to the antibiotic that's present. Could bring in some maths here. Okay, so area of a circle, pi r squared. And obviously radius, remember the r radius is half of the diameter. The larger that zone of inhibition, the more effective the antibiotic will be. Why would we use just a little filter paper disc soaked in distilled water as a control to allow us to compare those antibiotics to make sure it's not the filter paper causing that effect, for example? Look out for little hints and tips like that. Anything to do with cross-contamination, why we use tweezers and all of this side of things, you're all good on that. Okay, for just the GCSE biologists, we need to know about something called aseptic technique. Whole idea of aseptic technique is to avoid contaminating whatever your sample is with other microorganisms. So we're trying to keep our apparatus and our equipment, the area, as sterile as we can. This will be things like wiping down the desk before you do anything using a disinfectant, kills any microorganisms already present. Wearing gloves to avoid anything on your skin getting into your sample. It will be sterilizing glassware before we use it. It will be having a Bunsen burner flame close to where you're working, because that creates this updraft and therefore stops any of these microbes that may be in the air dropping onto the plate. If we're looking at how we're then going to transfer our bacteria from one place to another, we use a little wire loop. So sterilize it first of all, sticking it in that blue Bunsen flame, cool it, and then we can use it. And obviously we can make those little streak plates. So you sterilize your loop, take your sample, streak across one edge, sterilize your loop again, twist your dish, streak it across until you eventually just get a single colony that you can pick off. That means you can identify it through chemical tests, look at it under a microscope, etc. Back for everyone then, we need to know about medicines, and this is combined triple higher foundation, folks. We're almost there now. If we think about medication, generally these came from plants in the past, and what we have done is then use that knowledge to create synthetic versions in a lab. These days, what we can now do is just start with computer models. So we can have a little look, use a computer program and the very high power computers we've got to run little simulations based on what might be a good drug to use. Once we've identified that potential drug, we're going to go through our preclinical tests. Now, preclinical tests are basically the ones before a living thing, okay, like a human is involved. So we are going to start off with something like a tissue culture. So a little bunch of tissue or cells on a little plate. We could start with just bacterial cultures. The idea being here that if we use our drug on cells, tissue culture, etc., we're looking to see, has it just killed everything in that dish? If the answer is yes, we're not moving on from that point. Clearly it's not going to work. So we're looking for those big side effects at that point. If it passes the preclinical trial, we're then going to move on to the animal testing phase. We need to test it with 2 species at this point to have success. Animal testing allows us to see how it affects things like potentially behaviors, how it affects working systems. Does it mean that their heart rate shoots up ridiculously and makes them keel over? Does it stop them breathing? Those sorts of things we can only see with a living organism as an animal that a bacteria wouldn't show us. If we then have passed that one, we are moving on to the clinical trials. So testing on people. 3 stages to this. First one, healthy volunteers. We're looking for side effects at that point. We are purely going to give them our drug, look to see, do they spend their life sitting on a toilet after that? If the answer is yes, people may not want that drug, let's be honest. If we get minimal side effects, we are going to move on to the sample of volunteers with our condition, small number of them. That allows us to see how effective this treatment is. So we're now looking to see, is it having the desired impact on that condition? If it does seem to be working, what we then do is go to our large number of volunteers with the condition. Again, we're going to be going through looking for those potential side effects. We're going to be checking the effectiveness, seeing how good our new treatment is versus the old treatments that we may have. And that then lets us get to that end point of putting it on the market. A very lengthy and costly process that would be. We do need to know about something called the placebo effect, which is basically where people feel better because they expect to feel better. You could give them basically this little sugar pill that has 0 chemicals that are going to do anything of benefit, and because you're telling them this is a miracle drug that's going to make you feel all of these great things, some people will start to feel them because it's a psychological effect. The way we try to avoid that is using these things called double-blind trials, which are basically where neither the patient nor the doctor knows who's getting the placebo, who's getting the real drug. They will collate all of the data, and then it will be processed by an external agency who do know who's got which ones, and that then means we've removed that placebo effect from our results. We do need to know about some non-communicable diseases, nearly at the end now. First one is one of those lifestyle choices that hopefully none of you have made, which is smoking. Smoking, we need to know about 4 problematic parts of it. First one, tar. That's the stuff that's going to collect in the lungs and is carcinogenic. It causes cancer. That's why people who smoke have much higher rates of lung cancer. We then have nicotine. That's the addictive chemical. Also increases your heart rate. Carbon monoxide, as we know, this is going to bind to our hemoglobin irreversibly, thereby reducing our oxygen transport. People get out of breath really quickly. And then particulates, little tiny particles of solid that will end up in your lungs, engulfed by white blood cells, leading to this condition called emphysema. Lifestyle choice number 2 that could lead to some problems is alcohol consumption. So ethanol, which is in alcoholic drinks, is a depressant. It's going to slow down reactions. So short-term effects, just the typical ones that you see when people have got drunk. They lose balance, they fall down, they've got slower reaction times, their behavior changes. All of those are short-term. When they sober up, they return to normal, hopefully. Long-term effects, though, if you are using alcohol over a prolonged period of time and in reasonable amounts, then what we are going to find is you are more likely to get cirrhosis of the liver, which is where your healthy liver cells have basically become fibrous and fatty, and therefore your liver is not going to be as effective at removing toxins. We can get brain damage, heart disease, ulcers in the stomach, generally not good stuff. Moderation is the key when it comes to alcohol. Obviously, when you're over the age of 18, because clearly none of you would be drinking under that age because that would be illegal. We then come on to cardiovascular disease, which is basically any disease of the heart or blood vessels. One to know about is atherosclerosis, which is where your arteries have become narrowed and hardened due to deposits of cholesterol usually within them. Now, because of that, we can then obviously restrict blood flow to certain areas. Watch out for the ones going to the heart, so those coronary arteries, because if those ones are restricted, we're reducing the flow of blood to the heart. Therefore, it's going to get less glucose and less oxygen, which can lead to heart attacks. If it's the blood vessels going up to our brain that are being restricted, obviously, same effect, just with our brain cells, leading to a stroke in that region. Now, there are certain risk factors for cardiovascular disease, things like having a high salt diet. That's going to lead to more water being absorbed into your blood after filtration in your kidney, increasing your blood pressure. If you eat a high saturated fat diet, then more cholesterol gets deposited in the artery walls, narrowing them, increasing blood pressure there. If you don't exercise regularly, that's a downside as well. So one thing that we can do to reduce our risk of cardiovascular disease is stop smoking, stop drinking, take regular exercise, reduce the saturated fats, reduce the salt in our diet. All of those things are going to reduce your risk of CVD later on. So they're lifestyle changes we can make. If that's not going to cut it, then we can try medications. And what we will find is things like statins, most common one that are prescribed, which reduce your blood cholesterol. And that just stops it forming, basically. We've got these things called antiplatelets, reducing blood clotting because it stops the platelets being so sticky. Can potentially cause internal bleeding, though, because if the platelets aren't sticky, they're not clotting blood in a useful way either. Beta blockers, these ones are going to basically block the effects of adrenaline, slowing down your heart rate, also sometimes used in cases of anxiety as well. And then we've got our nitrates, which widen the blood vessels by relaxing the walls of them. Again, lowering blood pressure there. If the medication still doesn't cut it, then we need to go a little bit more extreme. We're going down the surgical pathway. We can replace valves if the valves are the problem. So if you're a leaky valve, blood is going to flow the wrong way. We can replace that either with a biological or a mechanical valve there. We can carry out a procedure called an angioplasty, which basically means we put this little thing called a stent in. It's kind of like a little mesh cage. We insert it into the artery, use a little balloon to inflate it, and basically it then pushes out on the walls, widening the artery once more. If that's still not good enough, we're going down the route of things like a coronary bypass, taking an artery from somewhere else in your body, like your leg most commonly, and then using it like a little bypass route. So if you've got a blockage here, we're going to then put a little loop going around it. So we're taking it around the blockage. If that's still not good enough, your last option is a heart transplant. You don't want a heart transplant. Try to avoid that one. There's a lack of hearts, and obviously you've got to match it to the recipient. Otherwise, we will reject it. These days, we are looking into things like stem cells, unspecialized cells from our Unit 1 work, remember, and we will usually use the embryonic stem cells because they can differentiate into any cell type. And they, if you remember, come from the spares from fertility treatment. We do also have another option of gene therapy, which is where we are going to replace a faulty allele with a functional allele. So this can be one of the treatments for cystic fibrosis, where we replace that faulty gene that's all to do with creating that thick, sticky mucus with a normal allele. Downside, it's only going to be short-lived, and there's no guarantee that that allele is going to get into the right place. Final thing to know about is the Human Genome Project, which is basically where we sequence the entire human genome. Once we did that, we could then identify the location of the genes that cause certain diseases, and hopefully that means we can develop drugs to target those particular alleles or proteins that they make. Do something to stop that particular problem because we know exactly where the disease-causing gene is. And that is the entirety of paper 2. So hopefully that has covered everything that you have needed for the papers tomorrow. Do obviously remember to read the questions very carefully. Don't leave any blank spaces. Blank spaces are just a guaranteed 0 mark. I guess worst case scenario, it'll be wrong. There's no negative marking. But best case scenario, you might stumble across the right word. You never know. Try your luck. So good luck tomorrow. And I will see you on the night before our next paper, which will be our chemistry paper, 6 o'clock again, to go through the entirety of paper 2 chemistry. So if you do have any questions, ping them in the chat and I will obviously answer those for you. Otherwise, good luck, get some sleep as well, and see you next time.